Well, good afternoon. The topic of today's lecture is going to be the lymphatic system and immunity. The lymphatic system includes the lymphoid organs and tissues, the lymphatic vessels, which contain a fluid called lymph, L-Y-M-P-H, and a population of individual cells called the lymphocytes. The cells, the tissues, the organs, and the vessels containing lymph perform three major functions in the body. The first is that the lymphoid organs and tissues serve as operating sites for the phagocytes and cells of the immune system that are going to provide the body with protection from pathogens. The second is that the lymphatic vessels containing lymph help to maintain blood volume in the cardiovascular system. And in addition to the maintenance of blood volume, we have fat absorption and the absorption of other substances from the digestive tract. And that's an important part of how fats are brought into the body from our diet. Thirdly, the lymphocytes are considered defensive specialists. They protect our body from pathogens like bacteria, viruses, but also from foreign tissue cells, which is one of the problems associated with tissue transplantation. The lymphocytes protect us from diseased or infected cells or cancer cells that have developed in our body that would otherwise pose a threat to the normal cells. And so in this chapter, we're going to talk about the lymphatic system and the components of the system and the functions of each of the components. We're going to discuss the importance of the lymphocytes and where you can find them throughout the body, as well as talk about the structure of the lymphoid tissues and organs and explain the functions of these tissues and organs. Lastly, we're going to talk about the immune system kind of from a broad perspective in terms of non-specific defenses as well as specific defenses or acquired immunity. So again, the lymphatic system is composed of lymphatic vessels that contain the lymph and the lymphoid tissues and organs. Let's see if this pen works here. Okay, so the lymphatic vessels and the lymphoid tissues and organs. Now, some of the functions of the lymphatic system include the transport of fluids back to the blood and an essential role in body defense and resistance to disease due to the presence of those cells called lymphocytes. Cytes meaning cells, cells in the lymphatic system that are playing a role in host defense. So lymph is itself a fluid. So one of the questions you may have is, how does it actually move? It's not blood, right? It's not moved by arteries and veins. So how does it move? Well, it moves through vessels called lymphatic vessels. And those vessels are a one-way system that move only towards the heart, right? So in the cardiovascular system, we know that there's a two-way system. Blood vessels move away from the heart, arteries, take the blood to the tissues, and blood vessels move back to the heart, veins. Right? So there's a two-way system. With lymph, it's a one-way system. There's no pump like the heart to move it through its vessel. And so the way lymph moves is actually through what we would call a milking action of skeletal muscle around the lymph vessels. So when the skeletal muscle contracts, it would be equivalent to, if you've ever seen a farmer milking a cow, putting pressure from the outside to move the fluid through the vessel. So we have a milking action of skeletal muscle, And we also have peristalsis that can occur in the walls of the lymph vessels due to the presence of smooth muscle. So if we were to look at the lymphatic pathways in the body, we start our journey with the smallest of all the ducts or the collection centers, which would be the lymphatic capillary. And the lymph is going to flow through that lymphatic capillary to a lymphatic vessel. Eventually, the fluid will move up to what we call a lymph node, and you may have heard this term before, the lymph nodes, and their importance in health. And you know, when a doctor is palpating your neck region and touching around your throat, he or she is trying to get an idea if your lymph nodes are swollen. But those are not the only lymph nodes in the body, and they're not the only ones that can be swollen, but they're often swollen during, for example, upper respiratory infections. But anyway, the nodes will drain into vessels, lymphatic vessels, which will drain into a lymphatic trunk, which will empty into a larger collecting duct, and eventually be dumped into a vein called the subclavian vein. Okay, so as you can see, the journey of lymph starts in capillaries and ends up in veins. The veins, therefore, take that lymph to the heart. So lymph is going to be delivered into one of two large ducts. The first is called the right lymphatic duct, and it'll drain the upper arm and the right side of the head and thorax. The upper right arm and the right side of the head and thorax. While the thoracic duct arises from the cisterna chyli and drains the rest of the body. Okay, so the thoracic duct will drain the rest of the body. And each empties lymph into the venous circulation at the junction of the internal jugular and subclavian veins on its own side of the body. Okay? So again, the lymph is being dumped into veins. And again, it helps maintain blood volume in that regard. It's going to move fat that we're digesting from the gut actually into the heart first, and then to the rest of the body. And so this is an important concept, and we're going to look at the wiring and how this is drawn up as we go. Okay, so again, if we're looking here at the thorax region, here we've got our major lymphatic trunks and ducts, and how they are connected to veins. So the blue color here, and I'm going to point this out, or maybe I could change the color of my, let's see, am I able to? No, not easily. All right, so I'm using the pencil in gray, so bear with it. But here we see the green, okay, are the lymphatic vessels. I did change the color somehow. And the blue are the veins. And so if you were to kind of put this into perspective, and if we start here, we see the esophagus, right, coming down, and the trachea is present. And so if we look, we have the left subclavian trunk, the left jugular trunk, and so this is where we're going to get lymph fluid draining into the blood vessels. Okay, we also see that if we look at the entrance of the thoracic duct into the vein here, okay, we're going to get lymph dumping into the vein. Okay, so what's important for you to see here is that whether we're on the left side of the body or on the right side of the body, right, this would be the right side of the body if we think about anatomical position. Okay, whether we're on the left or the right, ultimately the lymph drains right here where I'm pointing, and that is called the superior vena cava. The superior vena cava. Now, the superior vena cava is going to drain directly into the heart. Okay, so we can see that the lymph vessels are dumping into the venous network, and ultimately the place of entry into the cardiovascular system is going to be the superior vena cava, the large vein that comes down from the top of the heart into the heart. Okay, and so this other vein here that we're looking at, I'm going to point to the inferior vena cava. That also perfuses the heart, brings blood into the heart, and so the blood coming into the heart is happening through the vena cava. We have superior and inferior, but the lymph obviously is being dumped into there. So I don't expect you to be able to draw a line to everything on this picture, but if you were to kind of note some of the important blood vessels, obviously we have the subclavian vein and the superior vena cava as important components here. If we're to look a little closer at the lymphatic vessel, what we see is that they are present in and around the capillaries. Okay? And in this picture that I'm circling, they're depicted here by this green, looks like a reticular network. And so in fact, when the blood is leaving the artery and entering the capillary here, any fluid that leaks out can be collected by the lymph system. And eventually we know that lymph system is going to drain into veins. But basically the red is the artery. It's branching off into smaller arterioles and eventually into this capillary here that I've circled. Okay, it's giving up nutrients, giving up oxygen. The blood coming into the veins is depicted here as blue to symbolize deoxygenated. And any fluid that's lost in that interaction at the capillary will be collected back in through this lymphatic network. If you look here, this arrow is kind of blowing up one of the lymphatic vessels, and what you can see is that we have what we would call valves, or places into which the fluid can enter. And we see that around this lymph vessel, we have connective tissue, we have cells such as fibroblasts, we have connective tissue like fibers and proteins that are fixing this duct into the area where it is in the proximity of the capillary. And the actual lining of the vessel is made up of endothelial cells. Okay, so we have these lymphatic vessels in the proximity of capillaries, and basically their function is to pick up any lost blood fluid, any of the watery components, and eventually dump it back into the cardiovascular system to keep the blood volume relatively constant. Okay, so the lymphatic vessels, their purpose is to collect lymph from the lymph capillaries, to carry the lymph to and away from lymph nodes, and to return the fluid to circulatory veins near the heart. Are we good so far with that? And so the functions of lymph itself are several. I've already briefly mentioned the absorption of dietary fats, where fats are delivered to the bloodstream directly. So that's one important role. In the previous slide, we talked about the collection of excess interstitial fluid. Okay, excess interstitial fluid that may be coming out of the capillaries needs to be delivered back to the bloodstream. And then, if we have any kind of inflammation, remember, inflammation is going to require blood coming into the area to move those mobile inflammatory cells. So if there's any inflammation in the area due to a foreign particle like a bacteria or a protozoan, or you name it, any kind of infectious organism, the lymph will move those particles to the lymph nodes where we've got lots of lymphocytes that can help us deal with it. So the lymphatic capillaries are similar to blood capillaries, except they're very permeable, so that they can take up that cell debris, pathogens, or in some cases cancer cells. Remember, cancer cells can, or very frequently will, leave the main tissue and enter lymph, right? They metastasize to lymph nodes. So cancer cells can get in the lymph, and in the best situation, the lymphocytes will kill the cancer cells and prevent them from moving further and spreading around the body. And the cells that are going to make up those lymphatic capillaries are endothelial cells that overlap to form these one-way valves that let the interstitial fluid in. Interestingly, lymphatic capillaries are absent from bone, teeth, bone marrow, and the central nervous system. And there's a special type of lymph capillary called lacteals that are found in our GI tract, and as I mentioned, are involved with fat absorption. Lacteals are specialized lymph capillaries that are present in the intestinal mucosa. And so we're going to see in the digestion chapter that intestinal epithelial cells can have a large surface area due to the presence of these microvilli. And within the microvilli, we have these lacteals that allow for absorption of fat. So while most of the things that we absorb through oral ingestion, be it drug or food, are going to get into the blood and get taken to the liver, where the liver is going to act as a buffer to toxins and such, which we're going to learn about in the digestive chapter. Fat is actually going to not go that way. It's going to be absorbed through the lacteals, and as we saw, it's going to be dumped into the heart. Right? This is probably one of the functional reasons why high-fat diet leads to heart disease, because the fat gets dumped to the heart and can accumulate in the heart, and too much fat in the heart can result in problems. If we look a little bit more closer at the lymphatic capillaries here, we basically see that they're blind-ended tubes where we've got these endothelial cells that are overlapping with each other and are anchored down to the connective tissue around them. And they form these little one-way directional valves that let interstitial fluid in. So lymph is going to return certain materials back to the blood, like water, like blood cells, and like proteins, so that the consistency of the blood is maintained. But we've also mentioned that harmful materials can enter the lymph vessels and be present in lymph, things like microbes, cancer cells, or cellular debris. The lymph nodes are therefore going to filter lymph before it gets returned to the blood. Remember, the point of return to the blood is going to happen up towards the heart. Okay, so as the lymph flows from vessels into nodes, the nodes are going to filter that lymph and try to sterilize it so that things don't end up going to the heart and getting spread systemically. So luckily, within the lymph nodes, we have cells like macrophages, which are part of our nonspecific defenses, but they also play a role in cell-mediated, as you'll see later, defenses. And the lymphocytes, which are involved in the cell-mediated defenses. So one way to look at it is macrophages in the lymph nodes are involved both in nonspecific and specific immunity, while the lymphocytes are involved in what we're going to call specific immunity. Most lymph nodes are actually kidney-shaped and less than 1 inch long, and you can kind of feel them under your throat, in your throat region, under your chin if you feel around. Obviously, they get swollen during certain types of illnesses as an indicator of being sick. Now, the cortex of the lymph node, the outer portion, is made up of a collection of lymphocytes, while the middle portion, or the lymph node medulla, is primarily composed of phagocytic macrophages. Remember, phagocytosis is cell eating, right? Eating up debris or eating up bacteria that would otherwise like to spread around the body. So here's our typical diagram. Here's a diagram of a typical type of lymph node. What you'll notice first, I'm circling here, are the afferent vessels, meaning bringing lymph into the node. Okay, the lymph is coming into the node. And as you notice here, this structure is kind of designed so the lymph kind of has to flow all throughout it before it can make its way out of the node through an efferent lymphatic vesicle. Okay, so we have afferent vessels coming into the node, efferent vessels, I think I just said vesicle, but vessels, efferent vessels leaving the node, and in between we've got kind of like a roundabout type of journey that the lymph takes. And so what you'll see here is things called follicles within the lymph node, spaces within the lymph node, sinuses within the lymph node, and so within these what we'll call trabeculae, within these regions here, we're going to have lots of macrophages, and within the outer portions here, we're going to have lots of lymphocytes. Okay, so the slide you're looking at now shows you some of the many lymph nodes that are found throughout the body. And as you can see, there are definitely lymph nodes underneath your chin called the cervical nodes. But then there are lymph nodes all throughout the body, a lot of them in the thorax region, right, in the abdominal region, but also down our arms, in our armpit region, okay, and we have high concentration up here. So the thoracic cavity nodes, the axillary nodes, the inguinal nodes down by our reproductive organs, the pelvic cavity nodes, the abdominal cavity nodes, many types of lymph nodes, numerous lymph nodes in the body, all giving us defense and protection against tumor cells or invading pathogens. So you should feel comfortable knowing where an axillary lymph node is around your armpit compared to an inguinal lymph node down by your reproductive tract. Okay, and it just so happens that in most breast cancers that begin to metastasize, using that as an example, obviously the breast is located here in the pectoral region, and so very close by are the axillary nodes, which are usually the first site where a breast cancer may spread, just as an example. But again, we have these lymph nodes all throughout our body, and so when they become active, they can become swollen. Now, there are actually certain dedicated, what we call lymphoid organs in the body. And these sites are particularly important in terms of lymphocyte protection and function. The first of them is the spleen. The spleen is a blood filter, and it plays an important role. It actually destroys worn-out red blood cells. So, a typical red blood cell has a lifespan of 120 days, and as it works and as it goes through the circulatory system under all that pressure, it eventually wears out. It becomes what we call effete. And so effete red blood cells get destroyed in the spleen. The spleen also is involved in blood cell formation in the developing baby and acts as a blood reservoir. Okay, so the spleen is an important lymphoid organ. The thymus is located low in the throat. It overlays the heart, and it functions at its kind of peak levels during childhood. It produces a hormone like thymosin, which helps to program lymphocytes and is needed for the maturation of T cells. So the thymus is needed for mature T cells. The tonsils, they play a role in trapping and removing bacteria and other foreign materials. Tonsillitis, or inflammation of the tonsils, is usually due to a congestion of that tissue with bacteria. And lastly, we have lymphoid organs in the gut called Peyer's patches. Peyer's patches will capture and destroy bacteria in the intestine. All right, so you ate a contaminated piece of food that was filled with bacteria. You may never develop symptoms because the Peyer's patches are helping to protect your body from those pathogens. All right, so the spleen, the thymus, the tonsils, and the Peyer's patches are what we would call lymphoid organs. And so here's a photograph of the spleen in its normal position in the abdominal cavity. And this is an anterior view. Here you can see the spleen. You can see the diaphragm, right? So the spleen rests up against the diaphragm. You can see the adrenal glands here, very important gland in the sympathetic nervous system and the fight or flight response. Here you're peeking down at the left kidney. Big blood vessel here that supplies the spleen with blood called the splenic artery. And of course the pancreas, which is involved in digestion as well as in regulation of blood sugar, and intricately involved in, when it works well, protecting us from diabetes. Okay, so again, I'm not going to ask you to draw the spleen, but this is just a picture of where it sits in the body, kind of up against the diaphragm itself. Okay, so immunity. What is immunity? You know, the name itself, it refers to resistance to disease, being resistant to getting sick. Okay, so if you have a compromised immune system, you're very likely to get sick. On the other hand, if you have an immune system which is healthy, you're likely to fend off an attacking pathogen. And so immunity refers to resistance to disease. And there are two intrinsic systems that contribute to immunity: the innate or the non-specific immune system. Innate means you just have it. It's just there. It's innate, part of you. And innate just refers to from birth. You have it from birth. Adaptive is the other type of intrinsic system or specific defense system. So two major types: the innate immune system or the adaptive immune system. Sometimes that's called the non-specific immunity versus specific immunity. So just FYI, innate immunity involves things like macrophages, and I'm just going to abbreviate that here with the Greek symbol phi, macrophages. And adaptive immunity is going to involve cells like T cells and B cells, for example. So this slide kind of puts it all together. It talks about the innate defenses depicted for you here by this purple diagram versus the adaptive defenses. The innate defenses, things you're born with, include things like barriers. Right? We have barriers. We talked a lot about the skin, for example, in this class already. Right? Protecting us from our environment, from bacteria, viruses, or other things that would like to hurt us. We also have mucous membranes that line the respiratory tract, that line the nasal passages, that line the gut, that are going to trap pathogens or prevent physical injury by particles. Right? So these are just things that are innate, like you're born with them. There's nothing particularly specific about them. They're not different from one bacteria to a different virus. They're just there as surface barriers. Similarly, we have internal defenses like phagocytes and natural killer cells okay, which are intimately involved in destroying infection. Inflammation, the process of inflammation, which we've introduced in an earlier chapter, where we get increased blood flow to the site of injury or to the site of infection. It's going to bring in blood cells of the white type, white blood cells, that are going to be involved in resolving injury, right? Including phagocytes like neutrophils and macrophages, including the natural killer cells. There are certain antimicrobial proteins. One example would be lysozyme. And lysozyme is a protein found in various places, among others your saliva, but in other places within the body, that will cause lysis of foreign pathogens. Lastly, we have fever. Fever is an internal defense as well, which is innate. It's indicative of your immune system working hard to combat an infecting organism. Okay, so fever is an internal defense. By your body temperature getting hotter than normal, a microbe is presumably not going to grow as quickly, right? Because bacteria have a temperature at which they prefer to grow, which is your body temperature. But if your body temperature goes up by 2 or 3 degrees Celsius, which is pretty serious, the bacteria don't grow as well. So they're not growing as well. These cellular defenses are involved. Perhaps there's mucus trapping them. So you get all these kinds of cool innate defenses. In terms of adaptive defenses or specific defenses, then we have cells like B cells, which are going to make something called antibodies. So I would write that down somewhere, that B cells make antibodies. And T cells of various types, there's various types of T cells, that are going to be involved in cellular immunity. So again, the body has 2 defense systems for foreign materials, non-specific defense system, which are going to just protect against a variety of possible invaders. It's going to respond immediately to protect the body from foreign materials, while specific defenses are going to be required for each different type of pathogen. So you'll need a certain type of defense against a bacteria compared to against a virus compared to against a protozoan. Okay, and so that's a specific defense is required for each type of invader. It's also known as the specific immune system, or the acquired immune system. Acquired. Because you acquire immunity to each and every one of these single pathogens as a result of exposure and these different cells becoming activated. So again, the non-specific body defenses, things like the skin, the stomach mucosa, the saliva, and the lacrimal fluid, your tears that hydrate the surface of your eye, they contain lysozyme. So when you put your finger in your mouth, or you put your finger in your eye, that increases your risk of exposing yourself to a bacteria if you haven't washed your hands, but won't always result in an illness because of lysozyme. Of course, if you have a particularly pathogenic strain that you introduce to those tissues, or your immune system's a little bit run down, or you didn't wash your hands, then of course the mouth and the eye are sites of exposure to certain pathogens. But nonetheless, there's a level of protection there, the lysozyme. And of course, mucus. Mucus that lines your gut, mucus in your upper respiratory tract, and so on. So the defensive cells, who are they? Well, we've mentioned them, some of them at least by name, the natural killer cells. These cells lyse or kill viral infected cells and cancer cells, and they tend to be nonspecific because they use a type of agent to attack called a perforin. And that perforin punches holes into the membrane of the virus-infected cell or the cancer cell to kill it. So it's kind of like a missile shooter. It shoots missiles at the pathogen, those missiles being the perforin. Macrophages, we can have free or fixed macrophages. Fixed would be those which reside in the tissue. Free would be those which are going to come into the tissue due to inflammation. The neutrophils are phagocytes whose levels rise very shortly after exposure and then will fall after the pathogen is put under control. So the neutrophils are kind of like your first wave of phagocytes, followed by macrophages usually. The dendritic cells are defensive cells that will carry foreign bodies to lymph nodes so that lymphocytes can get involved and destroy the foreign particle or the foreign cells. So, a moment about phagocytosis, just a brief word about it. Phagocytosis, we call it cellular eating, and it actually involves certain steps. It involves a step called opsonization, which helps the phagocyte to adhere to the thing it's going to eat, whether it's a bacteria or a particle. It's going to engulf the bacteria or the particle by the process of phagocytosis, and then it's going to destroy that which it has eaten with lysosomal enzymes. Okay, this is not lysozyme per se, lysosome. Remember that organelle in the cell that is made up of lots of toxic things like proteases that can break things down. Okay, so lysosomal is not the same thing as lysozyme. That may be obvious, but I just feel like I should point that out. Lysosomal refers to the organelle within the macrophage, the toxic waste dump of the cell. So here we've got a macrophage depicted for you in purple, and it's using its cytoplasm to kind of extend and move itself around these invading particles. In this instance, the particles are bacterial cells. And you can see the macrophage is like grabbing onto them, right? We're in the very beginning of the endocytosis process, where we're grabbing on, we're adhering, and then we're going to internalize, and then those bacteria will be destroyed. So a few things about the innate defenses and inflammation. So right, if you look up here in the upper left corner, we've got a person who's put a nail into their skin, right? Maybe they were working with a nail gun, laying down tiles in the roof or putting down carpet or something. And so basically, this is going to be a type of injury that's going to immobilize your internal defenses, because now you've got the inside of your body exposed to the external, non-sterile environment. So here we've got injury, we've got inflammatory chemicals coming out of the inflamed site. Okay, so we've got release of chemicals, and those chemicals are going to, they have a special name, they're called chemokines. But for the moment, it's just important to know that there's chemicals released from the injured cell, which are going to recruit white blood cells to enter into this area. That's called leukocytosis, step 1 down here, where the neutrophils will enter the blood from the bone marrow. Right, so here they come. They've sensed that there's injury. They're coming towards the site of injury. But how do they actually leave the blood vessel and come into the site of injury? The way they do that is through a process called margination and diaphodesis. In margination, the neutrophils cling to the capillary wall. They stick very tightly. And in diaphodesis, they actually squeeze through the space in between endothelial cells. Right? So they stick tightly to the inside of the wall and they squeeze through the space between the endothelial cells. And then they continue to move towards the site of injury. And that movement is called chemotaxis, or movement towards a chemical signal. Right, so here they come, and they're going to start to eat up some of the cellular debris, some of the damage that's going on in the area. All right, so in terms of an injury, this could also be, a pocket of bacteria that's gotten into the skin. Maybe there was a scratch by a cat. It wasn't cleaned immediately, wasn't sterilized. Bacteria starting to grow down into the skin, a signal for injury. Right? So that nail could be a physical damage that tore up some cells, or it could be pathogens that have accessed the otherwise sterile environment. Okay, so leukocytosis, margination, diaphoresis, chemotaxis. So what are some of the chemical defenses that we have? Well, we heard about one of them from our guest speaker last week, interferons. Interferons are proteins that are secreted by virally infected cells that will go and move to healthy cells and bind those cells and cause those cells to become resistant to the virus. In fact, the name interferon, it was given that name because it's a protein that interferes with viral replication. Right? So we have interferons. In addition to interferons, we got something called complement proteins. Complement proteins. Those are 20 proteins in our blood that, when activated, can actually punch holes in infected cells and blow them up and destroy them. Complement proteins. Complement proteins float around in our blood. They're usually not active, but in response to an infection, they become active and they can destroy the cell that's been infected. And thirdly, we have chemicals called collectins, which are proteins that recognize foreign structures or sugar arrangements, and they stick to them. So the collectins are like a molecular signal. They see something foreign, they stick to it, and the immune system sees that they're sticking to something. They really shouldn't be sticking to anything if there's no infection. So they're sticking to something. That acts as a signal for the immune system. So interferons interfere with viral replication. Complement is a whole series of proteins that can destroy pathogen-infected cells and punch holes in them. And collectins are going to be, I don't know what you want to call them, tattletales. They're going to say, look, we've got a problem. They do that by actually binding to the surface of the infected cell and acting as a signal to the immune system. So if we look here, for example, at interferons, right, we've got two cells. We've got a cell over here. We've got a little spot by. We've got a cell over here. Right. In this cell, we've got an infection. A virus has come in, and it's replicated, and it's shedding, and it's releasing up here. It's releasing new viruses, and those new viruses would like to go to this next cell and infect it. But because the virus has entered the cell has a chemical defense called interferons. And as you heard last time at the guest lecture, there are many types of interferons, but the interferon gene will get activated in response to the virus infection. And the cell will start to make and release lots of interferons that will be able to travel to the nearby cell and turn on mechanisms that prevent that virus from being internalized. Okay, so if we start at step one over here, the virus enters the cell, but yeah, it's coming in and it's replicating and it's causing a problem, but by the same token, the cell is responding by producing interferons. And it produces them and releases them. And so the interferons that get released by that host cell are going to then bind to the surface of the adjacent cell, and that adjacent cell is going to turn on genes that are going to be used to prevent that virus from coming in, or prevent that virus from reproducing or replicating itself. So to simplify this, the infected host cell is making interferon. It's going to die and be killed by the virus, but the interferon that it released protects that second cell, that otherwise second cell that would be infected. It gets stimulated by interferons to synthesize genes that will defend it from further infection. Okay, so one example here of a chemical defense. Now, again, if we talk about the complement pathway, and I probably should tell you about this, even though it's a little bit more detailed, because I'm not sure where else you get it in your curriculum. So, there's two types of complement pathway. Remember, about 20 proteins would be considered complement proteins. We have the classical pathway and the alternative pathway. Now, in the classical pathway, we have something from the invading organism called the antigen that forms a complex with an antibody made by your B cell and certain complement proteins. Complement protein 1, complement protein 4, complement protein 2 are depicted here. That complex is going to coat the pathogen surface, and it's going to make phagocytosis easier for the neutrophils or the macrophages. Okay, that's the classical pathway. The alternative pathway is we've got spontaneous activation, stabilizing factors coming together, no inhibitors on the pathogen surface that are going to enhance inflammation. Okay, so they're going to come in, they're going to stimulate histamine release, there's going to be more blood coming into the tissue, more blood vessel dilation or permeability, phagocytes, chemotaxis. And so to make a long story short, complement proteins, to simplify this and maybe oversimplify this in some way, but basically there's two pathways, a classical pathway and alternative pathway that's going to lead to cell lysis, and the infected cell will die and not be able to perpetuate the virus or the I should say pathogen. So it's not just viruses, but also bacteria, yeast can activate these pathways. So for the purposes of now, just understanding that there are these proteins called complement proteins that are going to protect you from invaders. There are many of them. They can activate various pathways, one called a classical, one called an alternative. The net effect is slowing down the growth of the pathogen or killing the pathogen-infected cell. Fever. Remember, fever is elevating the body temperature, and that's going to be protective. Why is it protective? Because high temperatures of the body will inhibit release of iron and zinc from the liver and spleen, which bacteria would use for their metabolism. Right? So the bacteria aren't getting things like iron and zinc. In some cases, bacteria aren't growing as well because just increasing the temperature by 1 or 2 degrees slows down their growth dramatically. Okay, just like you don't feel good when your body temperature is elevated, many bacteria don't feel good if their body temperature is elevated. Fever also increases the speed of tissue repair, and we could discuss that point, but the cytokines that get released during inflammation cause increased vascular permeability. More of the blood cells can come in, respond to the infection, resolve the infection. And so fever is caused by a class of molecule released from macrophage called a pyrogen. Pyro meaning fire, gen meaning synthesizing. Pyrogen is a macrophage released product that causes fever, only released from the macrophage if there's infection. We've talked about the inflammatory response, chemical signals involved in that response are things like histamine, and histamine is released in large amounts by basophils and mast cells. So tissue is injured, basophils, mast cells, two types of white blood cells releasing lots of histamine that's going to cause dilation of blood vessels, increased permeability of the capillaries, and now things like neutrophils are going to move into the site of injury. So if you were to follow this flow chart from the top down, we see injurious agents causing our cells to get damaged, causing the release of cytokines, a molecule like histamine that's going to trigger increased vascular permeability. The blood vessels are going to dilate, they're going to become leaky. Those cytokines are going to cause neutrophils and then monocytes, which are a type of macrophage, to come into the area of damage. Right, so the blood vessels dilating, becoming leaky, is going to cause redness and heat. Right, the tissue is going to feel hot to the touch. The swelling of the capillaries and the leakiness is going to cause swelling into the tissue. That's called edema, and that's going to put pressure on the nerves in the tissue and cause pain. It may limit the movement of the tissue or of the joint, for example. And so the neutrophils are going to come in. Maybe the other white blood cells, like the monocytes, who are going to differentiate into macrophages, are going to remove the dead or damaged tissue. Okay, and so this is going to all contribute to the process of healing. Now, turning our attention over to specific immunity. Now, everything we've talked about thus far is non-specific immunity. Well, here we have a pathogen-specific response. Something about the pathogen is being recognized by the immune system. A little piece of the pathogen, a small molecule, like a molecular fingerprint or molecular signature, is called an antigen. Right, so the body might not have eyes to see that something has come in and infected it, but it can sense surface molecules on various things like bacteria. And parts of those surface molecules that activate the immune system are called antigens. They're said to be antigenic. So specific immunity is going to be pathogen specific. Your immune system might recognize a small portion of a surface molecule of a Mycobacterium tuberculosis compared to a small portion of a molecule on the surface of E. coli. Right? Those two bacteria, both bacteria, but they're going to elicit very different types of antibody formation by your immune system. Right? So without getting too complicated, let's just say that each pathogen has its own molecular fingerprint called an antigen, and so the immunity is specific to that antigen. When we see that signature or that motif or that antigen, we know that there's a certain pathogen in the body. Okay, so it's antigen specific, it's systemic, right? It involves the whole body response, the systemic circulation. And we say it has memory because usually subsequent exposure results in less illness because of the formation of memory cells that can make antibodies. Right, so maybe the first time you get a virus, you feel really sick and you're kind of out of it for 5 or 6 days. But if you were to be exposed to that virus 2 or 3 years later, you might not even be sick at all because your immune system has developed memory to those antigens and very quickly now has been able to eliminate the virus the second time or the third time around. Now, in order for the immune system to be specific, it has to distinguish self, you, from non-self invading pathogens. When the immune system fails to distinguish self from non-self, you start to attack your own body. Those are called autoimmune diseases. But I digress. So there's 2 types of specific immunity that we can talk about. Humoral, which means fluid, and that's going to be mediated by the antibodies that get secreted into the blood by B cells. And then cellular immunity, which is mediated by cells, in particular T cells. So humoral and cellular immunity are 2 arms of the specific response, or the specific immune response. Allergies. Allergies are not antigenic alone, but link up with our own proteins. What do I mean by that? I didn't really say that correctly. Okay. Allergies are caused by something called haptens, which are not necessarily on their own going to elicit an immune response, but when it mixes up with some of your own protein, it forms like a novel antigen that your body responds to. So, for example, hay fever, right? Being exposed to pollen. The pollen may be what we call an incomplete antigen, but when it interacts with proteins in your nose, when you breathe it in, the interaction forms an antigen that your body now responds to. So, if you remove the pollen, you don't have this hapten interacting with your own protein to form a true bona fide antigen. Haptens are an interesting topic, and I think they're a little bit more complicated than what I want to get across today, because sometimes you actually use haptens in vaccines to get a stronger immune response. So maybe for the moment we can put that one aside. And so let me keep going down to the cells here, right? So we have humoral immunity, we have cell-mediated immunity. Both types are mediated by lymphocytes, but there are different types of lymphocytes. B lymphocytes, T lymphocytes. Both types actually originate from hematopoietic precursors in the red bone marrow. Okay, so they come to us from bone marrow. So this explains why people who take drugs that suppress bone marrow get infections, like cancer chemotherapy drugs. Certain types of cancer chemotherapy drugs can hurt your bone marrow and increase the patient's risk of infection. So the cells of specific immunity are lymphocytes, B and T cells. B cells make antibodies, T cells are involved in cell-mediated immunity. Now this slide talks a little bit about the antigen receptors on lymphocytes, because lymphocytes are detecting antigens, right? Antigens, something foreign in the body. Right? And so there are receptors for those antigens on their surface. And what's pretty amazing, and again, this might be something you cover more in a full-blown immunology class, but what's really amazing here is that lymphocytes make up to 1 billion, and that is a B, different types of antigen receptors. So lymphocytes have the ability to make lots and lots and lots of different antigen receptors. And without getting into the genetics, there's something called somatic recombination that allows them to do that. Okay, so that's what we mean when we talk about antigen receptor diversity. And that's all I'm going to say about that. Now, each antigen will have a particular molecular shape that will stimulate a B cell to secrete antibodies to interact specifically with that particular shape, or that epitope. And so the B and the T cells recognize specific antigens through their plasma membrane-bound antigen receptor. And at any given time, a single lymphocyte can bear 100,000 receptors for an antigen that all have similar specificity. Okay, and so although it may encounter a large set of B cells and T cells, a microorganism is only going to interact with the lymphocyte that has the receptor for its antigen. Right, so you're going to have thousands of B cells and T cells floating around in your blood at any given time, but the one that's going to start the cell-mediated response is going to be the one that interacts the right receptor on the B cell or on the T cell. So in the world of B cells, we can kind of categorize them into two broad classes. The plasma cells. The plasma cells are those which are going to secrete antibody molecules at the rate of up to 2,000 molecules per second over the cell's 4 to 5 day lifespan. So the plasma cells are big-time antibody producers. Now, let's say that the infection has subsided. The body will retain some of the plasma cells, but then get rid of some of the others, the ones that are no longer needed. So the cells that stay develop into what we call memory cells. So now, if that memory cell encounters a similar antigen, it's ready to go. There's no kind of scaling up the process. The memory cell can quickly proliferate into many memory cells and eliminate the epitope of the, or bind to the epitope of the antigen. And so the reason why that can happen is called clonal selection. The first encounter is called the primary immune response. It usually takes longer than the second and subsequent encounters, which are called secondary immune responses, which are usually very quick, and a patient may not even experience symptoms. Okay, so the secondary immune response is stronger than the first. The response is much faster, much larger, and more prolonged. And so this secondary immune response we call immunologic memory, and it's the basis of vaccines. And again, we'll briefly discuss this in our other class in public health this semester when we talk about vaccines. So do you feel comfortable knowing the difference between a primary immune response and a secondary immune response? If you do, let's keep going. If you don't, just go back and think about that again before we move forward, because that's an important concept. So here's what the process looks like in terms of pictures. So here we've got our primary response up here in the upper left, the initial encounter with an antigen. So these little red balls would be the antigen. And the antigen binds to a receptor on a specific B cell. Okay, those B cells that don't have that particular receptor, so remember we said there might be 100,000 B cells and only one or two may have the right receptor for the antigen. But that's good enough, because once that receptor is bound, that cell starts to proliferate and clonally expand. When we say clonally expand, we say we're expanding the exact same cell. We're making copies and copies of it, a clone of the original. And they are going to expand into plasma cells. The plasma cells are going to make antibodies that are going to bind to these antigens on the invading organism. And so most of the plasma cells that get formed are going to be used to make antibodies. But some of the plasma cells will be retained as a memory cell. And that memory cell will be there for the second time there's an infection. Okay, so those antibodies that get secreted are big time important, because they help neutralize the invading pathogen. Does that make sense? So we start with lots of B cells. Only one might have the right receptor for that antigen, but once it's stimulated, it starts to expand. It proliferates into lots of cells we call plasma cells, and then usually a small but significant quantity of those plasma cells are retained as memory cells for secondary infection detection, and they form into memory B cells. Okay, so your immune system has memory. And so if we were to look at a graph here of the antibody concentration versus time after exposure to an antigen, we'll see that it might take anywhere from 7 to 14 days, and really 14 days or so, to make a lot of antibodies to the antigen. And then those antibodies are going to help resolve the illness, and then they will be scaled down in their production. We'll retain a few memory cells, and that's about it. Now the person's been exposed a second time to the same antigen, and boom, their antibody formation is much faster. You know, usually within a week, they've already made much more antibodies, and the immune system is much more competent in fighting that infection the second time. Let's imagine, though, at this point, in addition to being exposed back to that first pathogen, they get exposed to a second. Somebody else sneezed on them that had a different type of virus. Well, immunity to that second virus is going to follow the same path as the original. It's going to take anywhere from 7 to 14 days to produce antibodies, and then multiple days to resolve. And so upon second exposure to that second antigen, it would look more like this graph because of the memory cell. Does that make sense? Any questions? Okay. Coming to you live from St. John's campus in Rome. So the cell-mediated immunity. We have B and T cells that are involved, and they mature in the bone marrow and in the thymus. And their antigen receptors are tested in those tissues for potential self-reactivity. Okay, we don't want to be sending out cells that have an ability to destroy things if the things they're going to destroy are us and our tissues, because that would lead to injury. And so in situations where there's a failure of self-tolerance, we can say that that's a scenario where we can develop autoimmune disease, where T cells and B cells get produced that react against our own antigens, our own epitopes. Our own epitopes shouldn't be immunogenic, but they are being immunogenic, and they're causing an immune response. So T cells do have a crucial interaction with one important group of native molecules, whereas B cells do not. And what is that group of native molecules? So B cells, think of them as the antibody makers. T cells are going to interact with a group surface marker called MHC, Major Histocompatibility Complex. And so these are cell surface proteins that kind of give your cell its own signature, right? All of the cells in our body have cell surface proteins that are dependent upon our genetics. They're called major histocompatibility complexes. So T cells are looking for these MHCs, and when they see one that's foreign, they get activated, the idea there being, and the problem arising when T cells start to react against the host's own MHCs. And again, I don't want to go into this too much in terms of what is autoimmune disease, because that will be a topic you learn about in pathology. But the idea here is what's normally happening. Normally the B cells and the T cells are maturing in the bone marrow and the thymus, and they're being quality control tested before being put out into the circulation. And usually the standard is that if they're going to react against self, they'll be destroyed, so that they never have to go out into the systemic circulation and cause problems. But problems can occur if that was to happen, right? So failure of self-tolerance. Again, I think I've said all I need to say about that right now. You will revisit this in a future course. I'm not going to go into the MHC markers for the moment. I will tell you this: the MHC molecules are one of the reasons why organ transplants fail from person B to person A. That's why tissue matching is important. So if you got somebody from your family who can donate a kidney to you, it's much more likely that their surface molecules are closer to yours than a totally unrelated person. But nonetheless, when they look for a match, when they say I'm looking for a match, if I'm looking for somebody who's got similar MHC markers on their cells to you, so that the immune system won't pick up on the foreign MHCs and destroy them. So you'll learn about this in pathology again. I don't want to belabor the point, but MHCs are one of the reasons why tissue transplants can fail, because if they're too different from your own MHC structures on your own cells, then your immune system can detect them as foreign and destroy them. It's one of the reasons actually why people who get organ transplants are put on immunosuppressive therapy to reduce the risk of tissue rejection. The other thing I can tell you on this slide that might be new in this discussion is that there are different types of T cells. So you know how we said we have B cells, we have plasma cells, and memory cells? Well, with T cells, we have various subtypes. We have helper T cells, which do a lot of the job, but we also have cytotoxic T cells. We also have memory T cells. And so we'll briefly talk about them. Okay, I'm going to skip this slide, and I'm going to skip this slide. I'm going to skip this slide. And I'm not skipping this because it's unimportant, but this idea of antigen presentation, of major histocompatibility complexes, I think is going to be better served after you've had a course in biochemistry and you know what these proteins are, you know, and the kinds of things that surface proteins are able to do. So let's keep going. So this slide is one that I'll just briefly talk about, and I'll talk about it in the context of cell-mediated immunity versus humoral immunity. Okay, and so again, without going into every single detail of the slide, we know that in the cell-mediated response, we're going to have T cells involved, and we're going to have B cells involved. The T cells can be of different types. It can be helper T cells, it can be cytotoxic T cells. But basically, what they do is they work together with macrophages over here. The helper T cells work with macrophages to activate the cytotoxic killer T cells and the B cells. And so basically what happens is the cytotoxic killer T cell is going to destroy the infected cell and kill it, and the B cell is going to make antibodies, and those antibodies are going to help in neutralizing the spread of the pathogen. So these responses are called cell-mediated responses because, first of all, they're specific. There needs to be an antigen, a specific antigen response, binding to a receptor on the surface of the T cell. Right? So it's not like neutrophils coming in. Anything that's different, they eat it up. Yum, yum, yum. Here, a specific antigen activates a specific receptor, and there are cellular responses that occur because of that receptor activation. Okay? So, without moving too deep into this, there's two different types of responses here. We have the T cells doing cell-mediated immunity where they attack the infected cell. We have the B cell making antibodies that neutralize antigen, to slow down the spread of the pathogen. Okay, so as an overview, we can see how we have humoral responses and cell-mediated responses, antigens coming into the body through injury or infection, getting eaten up by a macrophage. The macrophage is going to interact with a helper T cell, which is going to stimulate a cytotoxic T cell to do toxic things to the infected cell. It's going to stimulate the B cell to make plasma cells and antibodies. In both cases, we're going to retain some memory cells. And so the next time we're exposed, the immune response is much more quick and much more strong. Well, I don't want to say much more strong, but much more quick. More antibodies get made faster, and so usually the patient might not even experience symptoms. And that's really why we do vaccinations, to establish memory cells. So how can we acquire immunity? Right? How can we acquire immunity? Remember, we talked in public health about a disease called AIDS, acquired immunity deficiency syndrome. Right? So we know that if we don't have acquired immunity, we're in trouble. So how do we actually acquire immunity? Well, there are different ways, but they all involve exposure. Right? You need to be exposed to things. Now, how do you acquire knowledge? You get exposed to things. Right? How do you acquire immunity? You get exposed to things. So you can have naturally acquired immunity or artificially acquired immunity. Naturally acquired is infection or contact with pathogens. Artificially acquired is vaccination. And then we have active versus passive in both types. So active means like an active infection, right? It's making me acquire immunity. Passive immunity is actually something that happens to little babies, that they get antibodies from their mother during breastfeeding. The antibodies can provide infection protection for the nurturing infant during the breastfeeding process. So one of the good things about breastfeeding is that you actually give your child passive immunity. You give them extra protection from illness when you breastfeed, which is not part of the deal when you use formula. Okay, there is no antibodies in the formula. On the other hand, in terms of artificially acquired passive immunity would be something like injecting serum into a patient, like what they're doing with Ebola now. They're injecting people's serum from people who have survived into people who are infected, and that serum contains antibodies that neutralizes the virus and seems to be working. So without getting too complicated here, the main concepts are acquired immunity. How do you get immunity? Naturally or artificially. Naturally can be active or passive. You should know an example of each. Artificially can be active or passive, and you should know an example of each. Now, antibodies. These are Y-shaped proteins. They're completely specific, and they have many uses for immunity and for science research in general. They're also commonly referred to by their category in terms of what they are. They're immunoglobulin proteins, or IG, immunoglobulin proteins, immunoglobulin. They're secreted by B cells, carried in the blood plasma, capable of specifically binding to an antigen. And there are 5 main types made up of 4 amino acid chains linked by disulfide bonds. And so here's what I'm talking about. Okay, we have 5 chains, 2 heavy chains, 2 light chains, and a stem. Y-shaped, they look like the letter Y. This portion is what's going to bind to the epitopes and neutralize them. Okay, we have a heavy chain, a light chain, a constant chain. This is sometimes called the FC region, this portion down here, which acts as a ligand for a receptor on macrophages. Okay, so macrophages will eat these things up and break them down. Lots of disulfide bonds, which you're going to learn about in biochem, so I won't talk too much about them. The point here is the structure is important for the function. Like this thing acts like a clamp. It binds to the epitope, and then the bottom portion brings it to the macrophage for elimination or clearance. All right, just another picture of immunoglobulin structure, again without getting into too much detail at this point. Now there's 5 types of immunoglobulins, very important for immunity, all of them actually, but IgG is involved in defending against bacteria, viruses, and toxins. IgA is also involved in fighting against bacteria and viruses. And there's three other types: IgM, IgD, and IgE. IgE really important in allergic reactions. So these are immunoglobulins: IgA, IgD, E, G, and M. By far the most commonly used immunoglobulin clinically and therapeutically are IgGs. IgEs are involved in inflammation, so you want to keep that in mind. Promotes inflammation, allergic reactions. And IgMs are involved in things like complement activation, and they can react with antigens on red blood cells following certain blood transfusions. So, point be told, there are several types of immunoglobulins that that act as antibodies, and they have different functions. IgG is by far the one most associated with what we're talking about today in terms of fighting infection and eliminating infection. Okay, so these are the different types of immunoglobulins, along with a description of what they do. And I'm not going to hold you to all of the details beyond that for IgG at the moment. You're going to have a whole course on immunology, so I presume you're going to learn about all of the Igs. But here, IgG, very important. It crosses the placenta. It confers passive immunity onto the developing baby. It protects against bacteria, viruses, and toxins in the blood and lymph. And it triggers the action of the complement system. Okay, so very important immunoglobulin. And I'll leave it at that. Okay, so in your study, if you know what IgG is and what it does and its role, you've gotten what you need. But also just realize there are other types of immunoglobulins, and they all do certain things. Okay, and sometimes those things can be overactivation of the immune system, causing a severe allergic reaction, like IgE. All right, so here's our antigen, forming an antigen-antibody complex with the antibody, which is going to be inactivated. The antigen is going to be inactivated by neutralization by the antibody, by agglutination, so cells are going to come and bind around the antigen, by precipitation. Okay, so it might precipitate out lots of these microbes, and phagocytosis, clearing these things away. In addition to that, antigen-antibody complex activate complement pathway, and the complement pathway can lead to cell lysis, it can enhance phagocytosis, and can enhance inflammation. Okay, so the antibody is critical to all of these things. If the antibody is not there, these things don't happen. And hence, this idea of antigen-antibody complexes and effects on immunity. Now, grafts. In terms of transplants for tissues like a kidney, tissues like bone marrow, heart valve, what you will. We use terms to describe the types of transplants. We have isograft, which would be an identical twin with the identical surface molecules, major histocompatibility complexes. So this would be a bone marrow transplant from a healthy twin to a twin who has, for example, leukemia. That would be called an isograft. So be comfortable knowing what that term means compared to an autograft. An autograft is something where skin, for example, from one part of your body would be used to replace burned skin. So maybe a person has burned a bad burn on their, part of their face or part of their hand, and skin is used from another body region and grafted onto those burned sites. to promote the healing process. Autograft, grafting part of yourself from one place to another. Okay. Allograft. Allograft is from the same species. So a kidney transplant from a relative or a closely matched donor. Right? So maybe a first cousin has similar major histocompatibility complex gene as the person who needs the tissue. And how would they know that? They would do a molecular analysis. They would do a blood sample and look at the DNA and get an idea for how similar the tissue surface molecules may be. An allograft. Okay, so an allograft is when a first cousin or an uncle donates a tissue that gets put into the recipient. It's different from the self, it's different from an identical twin, but it's close. And last but not least is xenograft. Xeno means foreign. Xenograft is going to be a graft from a totally different species. So using, for example, a heart valve out of a pig's heart to replace a heart valve, a defective heart valve in a human. A xenograft. The term autoimmunity, self, is when the immune system does not distinguish between itself and foreign. And so the body makes antibodies and sensitized to lymphocytes that attacks itself. So it sees the joint, the synovial joint in rheumatoid arthritis as foreign, and it starts to attack the joint, causing pain and inflammation. Autoimmune disorders are numerous, and I'm not going to go through them all, but some of the big ones, like type 1 diabetes, is considered by some to be an autoimmune disease, where the pancreatic beta cell of the beta cell of the pancreas is destroyed by the host immune system through a mechanism that's not very clearly understood. Okay, so that's one kind of prevalent example. We see lots of cases of type 1 diabetes. Used to be called juvenile onset, but now we realize it's not just children anymore that can develop that. But usually, interestingly enough, develops after a viral infection when it happens. Okay, but if you look down the list, the other one I'll point out is rheumatoid arthritis, leading to joint pain or joint deformity. And usually, antibodies are developed against the cells that line the joint, the synovial cells, and so start to get damage to the joint. And as you know, the joint is well perfused, well innervated, and can cause, which leads to edema or inflammation of the joint. I should say really inflammation of the joint, not just edema, and pain. So be comfortable knowing those two examples of autoimmune disease and just realize that there are many others. Lastly, allergic reactions, and this is going to be really relevant for you as a pharmacist, future pharmacist, because you want to avoid these in your patient. And there are 4 different types, and again, I know you're going to go into the molecular mechanisms behind these in your immunology class, but just I would be amiss to overlook this. So there are 4 main types. Type 1, allergic reaction, is the most serious. This one can kill you. It's an immediate reaction allergy. It could present as hives, or it could present as anaphylactic shock, where a person has difficulty catching their breath, severe bronchoconstriction, laryngeal spasm, can die. So anaphylactic shock has been associated with certain drugs like penicillin in some patients, bee stings in some patients, serious things. Some patients that are allergic to these things actually carry around epinephrine injection, an injectable pen. If heaven forbid, they're far from medical help, they could at least inject themselves until medical help arrives. They're out camping or, so they're skiing or something. All right, so type 1 is the most severe. Type 2 is antibody-dependent cytotoxic reaction, usually going to develop over a few hours, and is often associated with blood transfusion reactions in some patients. Type 3 are immune complex reactions. This is where the antibody complexes that are formed can't be cleared from the body and can lead to damage, particularly of the kidneys. And type 4 are delayed reaction allergies, so they might develop, 1 to 3 weeks after an exposure. It might present as a skin rash or something to that extent. But by most standards, the type 1 would be considered the most serious. And so this is immediate reaction allergy. IgE antibodies are being stimulated on mast cells. Mast cells are dumping out lots of histamine, and this could be a medical emergency. And you could read a little bit about that, more detail about that a few slides back, where we talk about the different IgEs. But again, I'm not here to get into all the mechanism of this. Really, I want to give you the overview today of the lymphatics and the immune system. So hopefully, because we've gotten to the end of our slides here, hopefully you've developed an appreciation and an awareness of the relationship between lymphocytes and nonspecific cells. Talked a little bit about the anatomy of the lymphatic system, lymph vessels, lymph nodes. We talked about absorption of fat via lacteals and the importance of the lymphatics in that process. And then we talked about some of the lymphocytes, the key players in cell-mediated immunity or humoral immunity, like the B cells for antibody production and humoral immunity, and the T cells for cell-mediated immunity. So I'm very glad that I was able to record this for you. It's a very important topic, as I hopefully have showed you here. And of course, if there are any questions, I'm going to be available to try to help you through this material the best I can. So I will be giving you an assignment, like a quiz related to this information, which will probably be a homework quiz. And so if you have any difficulty, please let me know. Have a great day. I'll see you soon. How do I stop this? I have no idea. Exit? I hope it's recorded. I just spent a long time doing this. Let's see. Here goes.